Good afternoon and welcome to our webinar on Freeing Up Tech Time, Advances in Osmolality Testing. Today's webinar will be led by Julie McKenzie and Adam Leonard. Julie McKenzie is the Senior Manager for the Clinical Product Portfolio at Advanced Instruments. Her background is in biochemistry, cellular, and molecular biology. Adam Leonard is a Technical Product Manager with experience developing a range of surgical and diagnostic technologies. He joined Advanced Instruments earlier this year and along with Julie has led the commercialization of the new OsmoPRO MAX automated osmometer. We encourage you to ask questions in the question box that you can find on the right side of your screen. We will be answering 5 questions at the end of the webinar, and any questions we don't get to, we will respond to directly after the close of the webinar. Take it away, Julie.
Hi, I'm Julie McKenzie. I manage the clinical product portfolio at Advanced Instruments. I've been with the company for about 14 years now, and my background is in biochemistry. I'm really excited for today's webinar. There are several objectives for the webinar today. The first objective is to understand how osmolality testing is used in a variety of different patient scenarios. We'll explore use cases from hyponatremia to toxic alcohol ingestion and monitoring of therapeutic drugs. Second objective, you'll learn how automating osmolality testing in a hospital or reference lab setting of any size can help your team specifically when it comes to freeing up tech time and also improving result turnaround times, reducing errors, and ensuring compliance. Also during this webinar, you'll get to see a demonstration of our OsmoPRO MAX automated osmometer, which was specifically designed to free up tech time. Osmolality is a measure of concentration, unlike a test that is measuring a specific analyte. This makes osmolality an extremely powerful tool in patient care settings, the biotech and pharmaceutical industry, and even in food and beverage. Within the patient care setting, osmolality is a high-value, low-cost test ordered for numerous clinical applications. Osmolality provides valuable information to clinicians in many different specialties, from nephrology to emergency medicine and intensive care, about what's happening in the patient's body. Osmolality narrows the list of diagnoses from over 100 possibilities to fewer than a dozen, and it's measured using a freezing point osmometer. This method of osmometry is critical in patient care settings where patients may have ingested toxic alcohols or they may be receiving certain osmotically active therapeutic drugs. These substances are ones that may interfere with other methods of osmometry and deprive the clinician of valuable information needed to make treatment decisions. Let's take a look at 3 common use cases for osmolality testing. Let's discuss the critical role of osmolality testing in identifying the culprit behind an electrolyte disorder, specifically hyponatremia, which is low blood sodium. When we look at the clinical pathway for diagnosing hyponatremia, measurement of serum osmolality is the first step and helps determine if a patient has true hyponatremia or pseudohyponatremia. Once true hyponatremia is confirmed with a serum osm, urine osmolality then helps narrow down the causes. This is important because treatment of hyponatremia differs based on the cause. In fact, treatments may be opposite. In one case, the clinician may give fluids, while in another, the clinician may restrict fluids. Hyponatremia can be life-threatening, making the case for in-house osmolality testing to ensure result accuracy and timely turnaround. In fact, this is one reason that osmometers are a staple in many hospitals, and they are actually located at the same campus as the patients who are getting the testing. Osmolality is also a valuable tool in identifying alcohol intoxication. Here, we're looking at a case where a man has presented to the emergency room and is suspected of having ingested antifreeze. The clinician wants to identify the osmolalal gap. This is the difference between the patient's measured osmolality using an osmometer and the patient's calculated osmolality. A large positive osmolalal gap, in this case, helps confirm the presence of a toxic alcohol and inform treatment. This is a key case where calculated osmolality is just not sufficient. Measured osmolality is necessary to determine the osmolalal gap. Additionally, this is a case where freezing point depression is a must because alcohols interfere with other osmometry methods. Like hyponatremia in the case we just looked at, toxic alcohol ingestion can be life-threatening, again making the case for in-house osmolality testing to ensure result accuracy and timely treatment. The last case we'll look at is utilization of osmolality in monitoring therapeutic drugs. In this case, a woman is receiving mannitol therapy, an osmotically active therapy to decrease intraocular pressure after cataract surgery she had. As in the previous case, the clinician is interested in identifying the osmolar gap, which will provide insight into proper dosing. In this case, the large positive osmolar gap helps confirm mannitol overdosing. This is a case where freezing point depression is a must because mannitol is a sugar alcohol and interferes with other osmometry methods. Mannitol therapy requires close monitoring. In fact, osmolality is often ordered multiple times over the course of therapy, again demonstrating the importance of in-house osmolality testing to ensure result accuracy and timely decision-making. Now that we've had a chance to dive into some of the important use cases for osmolality testing, we'll bring it back to the lab. Our mission is to help laboratories quickly and confidently deliver accurate results to physicians to help guide treatment decisions. This is critical now more than ever in this environment where we're seeing unprecedented staffing challenges. We offer several products to help tackle these challenges and help labs improve workflow efficiencies. One of these products, the bottom left of the slide, the glow site, helps eliminate the manual hemocytometer method by automating CSF cell counts down to 0 cells per microliter. Another, the Inox mat, is an anaerobic jar system that saves labs time by creating consistent and reliable environments for growing bacteria in under 5 minutes. We also provide a neonatal bilirubinometer that provides both total and direct bilirubin results in 2 minutes. In this webinar, we'll discuss how our osmometers, and specifically the OsmoPRO MAX automated osmometer in the top left of the slide, helps labs improve workflow efficiencies. We developed OsmoPRO MAX in close collaboration with hospital laboratories around the world. We really listened to the challenges that you're facing and worked hard to deliver a solution that would free up tech time, deliver faster result turnaround times, improve accuracy through automation, reduce the risk of errors, and ensure ease of compliance. For us, it's about taking the burden off of your team and placing it on the instrument where it belongs. With OsmoPRO MAX, we made automation possible within a small footprint, so it's attainable for all labs. There are no plastic test cups or tips with the OsmoPRO MAX and no chamber cleaners or wiper rings. Techs of all skill levels can now simply place patient samples onto the instrument, press start, and walk away. The instrument accommodates a variety of tube sizes, including primary tubes. If user ID is set as required by the supervisor, users will be prompted to input their IDs or scan their badges. The system automatically scans the patient barcode, eliminating transcription errors, and replicate testing is programmable, so users don't need to stand at the instrument to run controller patient replicates. Once a result is ready, it can be manually or automatically sent to the LIS, eliminating paperwork and time spent manually inputting results. Similarly, QC data can be sent directly from the device to the Advanced QC Peer Group program. User intervention is limited to loading and unloading samples and replacing a bottle of system fluid after 500 tests. This slide highlights several key features of the OsmoPRO MAX. I'll touch on just a few that we haven't covered yet. The first is continuous loading of samples. Continuous loading in the sense that you don't have to manually intervene between samples to clean or remove test cups, and also continuous loading in the sense that you can easily add both routine and stat samples to the queue when the instrument is already testing other samples. OsmoPRO MAX helps labs like yours maximize resources. Sample volume no longer dictates whether we point you towards a single sample or multi-sample osmometer. Everyone deserves automation, whether you're testing many samples a day or just a few. OsmoPRO MAX can be used as a single sample or multi-sample automated osmometer. It enables your staff to get time back, opens osmolality testing up to techs of all skill levels, and improves result turnaround times to help inform timely treatment decisions and minimize patient length of stay to the extent possible. With that, it's time to see the OsmoPRO MAX in action. My colleague Adam Leonard will be demonstrating the OsmoPRO MAX for us. Over to you, Adam.
Thanks, Julie. So now I'm going to explain how labs of all sizes can gain greater efficiency through automation with the OsmoPRO MAX. Unlike other osmometers, the OsmoPRO MAX utilizes flow-through technology that eliminates manual pipetting and consumables. The freezing of samples occurs in a cooling module within the instrument, so there's no longer a need for sample cups or sampler tips. Sample pipetting, testing, and cleaning are all performed automatically, minimizing the amount of time that needs to be spent at the device. And continuous loading and unloading allows samples to be tested as soon as they arrive in the lab, removing the need to batch test. Let's start by walking through some of the key components of the system. So system fluid is used to clean the fluid lines after each test. The volume in the system fluid bottle is sufficient for testing 500 samples. There are two peri pumps located on the side of the instrument that are used to move samples and system fluid through the system. A waste bottle then collects used system fluid and tested samples. After testing 500 samples, the items in this maintenance kit would be replaced. The kit contains a bottle of system fluid, a vial of enzymatic cleaner, and two replacement peri-pump tubing lines. Onboard animations guide the user through how to replace each item in the kit. So no daily cleaning and maintenance is required with the OsmoPRO MAX, just the kit replacement every 500 tests. Since there are no consumables and no manual cleaning steps to perform after each test, samples can be continuously loaded and unloaded from the 20-position turntable. To eliminate manual pipetting, a kit of turntable adapters is included with the instrument. These enable automation of the QC and calibration procedures. The kit includes vial adapters for each level of our pro-null and re-null controls, and ampule adapters for our cleartrol 290 reference solution, calibration standards, and the vial of enzymatic cleaner that's included in the maintenance kit. To run a test of our cleartrol 290 reference solution, first you would open the ampule, you would then place it into the ampule adapter, and then place the ampule adapter onto the turntable. The adapter is barcoded, so the system will recognize that cleartrol 290 is in that position. If you're using our pro-null and re-null osmolality controls, the vials for each level can be placed into their respective adapters and then placed on the turntable. And this allows you to fully automate the QC process, saving a significant amount of time each day compared to other osmometers. I'm going to now give a quick overview of the user interface. On the left, the 20 positions shown correspond to the 20 positions on the turntable. Positions with sample tubes that have been scanned will appear in gray when added to the testing queue. A position will turn orange when a test is in progress, followed by green when testing of all replicates for that sample is complete. The OsmoPRO MAX allows for automated replicate testing with the number of replicates configurable for both patient samples and controls. At the top right of the screen is the status bar. The bar will turn orange when a test is in progress with the test completion percentage displayed. Below the status bar is the results table. Here you will see the position, result, and sample ID shown. In addition, our results database stores the prior 1,000 results on the system. At the lower right are the navigation options. Currently, the user can either go to the menu or select the start button to begin testing. When a test is in progress, additional navigation options will be presented to allow the user to add new samples to the turntable. On the lower left are the system status indicators. Furthest to the left is the system fluid level indicator. The indicator is currently green, with the number above it showing the remaining number of tests in that bottle of system fluid. The indicator will turn orange when there are fewer than 100 tests remaining, and red when there are fewer than 25 tests remaining. So the user will be notified when it is time to replace the maintenance kit components. Next to the system fluid icon is the QC status indicator. This is a feature unique to the OsmoPRO MAX that checks the results database to see when the last successful Clear-12 290 test was run. The unit can be set up to lock the system if Clear-12 290 has not been successfully run in either the past 8 or 24 hours. A successful test of Clear-12 290 or a supervisor override would then be required to unlock the system. This feature can help prevent patient samples from being run if daily QC has not been performed. Lastly, the LAN icon indicates the status of the connection to the laboratory information system if the unit is interfaced. So now that we've reviewed the user interface, I'll show you how to run samples on the OsmoPRO MAX. Primary sample tubes can simply be placed on any open position on the turntable. The turntable accommodates a wide range of tube sizes, ranging from 11 to 17 millimeters in diameter and up to 100 millimeters in height. As far as the minimum sample volumes, approximately 450 microliters is required when testing directly from a primary sample tube. This will vary slightly depending on the tube dimensions. If you do receive a sample with a volume less than 450 microliters, we do offer small volume inserts. And when using these, the minimum sample volume required will be reduced to 170 microliters approximately. Once all samples are loaded, all the user needs to do is to press start. The user can now walk away from the instrument. An integrated barcode scanner located at the back of the turntable automatically scans each tube sample ID, mitigating the risk of sample mix-ups and reducing the potential for transcription errors. The instrument will then automatically pipette the sample directly from the primary sample tube. The sample will travel into a cooling module located within the instrument, and that's where the freezing occurs. When not in use, the automatic pipette is stored safely within the instrument. Going back to the home screen, so position 1 has now been scanned in, and that position is currently being tested. And that's what you're seeing here in orange. The system is configured to automatically run 2 replicates per position. So we see position 1-1, position 1-2, position 2-1, and position 2-2, along with the sample IDs for both positions. Now that a test is in progress, there are 2 new navigation options, Load More and Stat. To add a new sample to the turntable when a test is in progress, all you need to do is to push one of those buttons, select the position where you've added the new sample, and press Start. The user does not need to wait for the current test to be complete in order to load a new sample. The sample that was just added will be scanned into the system once the current test is complete. It will either be added to the end of the testing queue or the beginning, depending on whether Load More or Stat was selected. The process for running daily QC is similar. The Clear-12 290 reference solution and pronull and renal osmolality control adapters are loaded onto the turntable. The number of replicates to be tested is configured in the settings menu, and all that needs to be done at that point is to press Start to begin testing. A new feature with the OsmoPRO MAX is that the system can be configured to automatically transfer quality control data for pro non-renal to the Advanced QC Peer Group program. This eliminates the need to manually transcribe results, saving both time and preventing errors. Moving on to calibration, this process will be infrequent with the OsmoPRO MAX. The instrument arrives factory calibrated, with recalibration only expected to be performed approximately once every 6 months. All levels required for calibration are included in the calibration kit. So we have the 0, 300, 850, and 2000 milliosmol levels, as well as an ampule opener. In order to run the calibration, the ampule adapters for each one of those levels would just need to be placed onto the turntable, and then the calibration process can be completely automated. By automating osmolality testing with the OsmoPRO MAX, we feel that labs of all sizes can minimize errors, reduce turnaround times, and free up staff to focus on other priorities in the lab. I hope you enjoyed the demonstration. Now I'll turn it back over to the moderator for the Q&A.
Thanks, Adam and Julie. The first question is the OsmoPRO MAX currently available?
I can take that question. This is Julie. So yes, the OsmoPRO MAX is available globally. Currently, we're taking orders with shipments beginning in early December. It's crazy to think that December is almost here. There is a 4 to 6 week lead time with the potential for expedited shipping.
Great, thanks. The second question is, does the OsmoPRO MAX interface with the LIS?
So yes, the OsmoPRO MAX does interface with the LIS. On the back of the device, there is Ethernet connectivity, and via middleware, the instrument, the OsmoPRO MAX, has bidirectional connectivity. One example of a driver that's available is through data innovation. So those using Epic, Instrument Manager, if you have an open port, you'll be able to get that all hooked up.
Great. The third question is, our lab is currently using protanol and renol for QC, but we didn't realize Advanced Instruments has a peer group program. How can we sign up?
So really easy and quick to sign up for the Advanced QC Peer Group program. So one of the ways to register, you can go to our website for the peer group. So it's aicompanies, that's plural.com/advanced-QC. And I'll repeat that one more time. So it's aicompanies, which is plural.com/advanced-QC.
The fourth question is, can I schedule an OsmoPRO MAX demo for my lab?
Absolutely. We are happy to provide virtual demos. You can request a virtual demo also by going to our website. If you go to aicompanies.com/OsmoPRO MAX, on the right side of the screen, there's a request a virtual demo tab, which will allow you to make that request, and then someone will be in touch shortly to schedule a virtual demo for your team.
Perfect. Third question is, will a recording of the webinar be available? And I can take that one. Yes, we will be hosting a recording of the webinar on our website after the close of this webinar. And once it goes live, we will be sharing the link with all of you so that you can distribute out the webinar as you see fit and as is helpful for you and your team. Now we'd like to ask a few poll questions. You'll see them pop up on your screen. Thank you so much for taking the time to join us today. We hope you've enjoyed the webinar. There were some questions that we didn't get to, so we will be reaching out after to answer those for you, and also feel free to schedule time to discuss with us in more detail if you have any specific questions or concerns about your lab and how the OsmoPRO MAX could help streamline your workflows.